and welcome to our video lecture on topic 2.5 enzymes, our essential understanding for today. Enzymes have an active site to which substrates bind. We're going to talk about what are active sites and what are substrates. On the screen right here, we have a wide variety of some proteins. Remember our proteins from our last unit: rubisco and insulin, rhodopsin and collagen, spider silk. We are definitely going to be focusing on proteins like rubisco, our enzymes. Our objectives for today include a couple pieces of protein review, since enzymes are mostly proteins. Ribosomes are special kinds of catalysts that are composed of protein and RNA. Enzymes are catalysts. A catalyst is a substance that speeds up chemical reactions by lowering the activation energy. We're going to talk about how proteins can be denatured. We're going to talk about how the function of a protein is related to its structure. We're going to focus on enzyme structures. We're going to look at enzymes as metabolic proteins. We're going to talk about how they have active sites to which those substrates bind. We're going to define active site and substrate once again. The rate of, that should say of, not if, the rate of enzyme activity can be altered by substrate concentration, temperature, pH, and inhibitors. And we're also going to look at a few uses of enzymes in technology. Remember that there are lots of functions of proteins. We are going to focus on enzymes. Rubisco is an example of a protein used by plants. Catalase is an enzyme that breaks down hydrogen peroxide. Amylase breaks down amylose. Pretty much anything that ends in an ase is going to be an enzyme, and these are all about metabolism, causing chemical reactions to occur in biological systems. Enzymes have active sites. Active sites are what those substrates bind to. We have a couple different models of how our substrates bind to active sites. There's one idea called the lock and key, where we have the substrate. The substrate is the thing that the enzyme acts on, and its shape is a perfect fit for the shape of the active site of the enzyme. Those guys just kind of slide in together, and then things can happen. The enzyme acts on the substrate, and then we end up with whatever products there are. A better model is this idea of induced fit, where we don't have a perfect shape fit for the substrate and the active site of the enzyme, but instead what happens is when the substrate goes into the active site, it causes a shape change in the enzyme, and we end up with this enzyme substrate complex. That shape change helps to make the enzyme have its action on the substrate. Both of these models do help us understand enzyme substrate specificity. Enzymes and substrates are specific to each other. Enzymes can't act on any old substrate. Substrates can't fit into any old enzyme. There does have to be some kind of fit, whether that be lock and key or more likely induced fit, that leads to the specificity. These illustrations are really nice to show different ways that an enzyme can act on a substrate. So here we have a catabolic reaction, where the catabolism, we're cutting things or breaking down bigger molecules into smaller molecules. We have our substrate here. It looks like it might be a disaccharide, and it's binding to the active site of the enzyme. The enzyme's active site is that piece that fits quite nicely the shape of the substrate. Once we have that complex enzyme substrate complex, the enzyme is putting some kind of strain on the bonds, which helps to break that bond with a little bit less energy than would be required otherwise. We end up with our products, and this is glucose and glucose, which means that this guy must have been, hopefully we remember that two glucose molecules bound together in a disaccharide is maltose. And so here we have our two pieces of glucose separate. This was catabolic. We broke that maltose down into glucose. Notice that we had to add some water because we know this reaction is hydrolysis. Good? Good. Notice that the enzyme here exits the reaction. It is totally able to carry out another reaction. It is not consumed during the chemical process here. The opposite of catabolism is anabolism, and we have a super similar thing happening, but instead of having one thing that's broken into multiples, we actually have two substrates, so our two glucose monomers, binding to the enzyme's active site. We're going to release some water because we know that this is condensation. And the enzyme is actually going to help to align those two pieces so that it is easier to create a new chemical bond between them. And we end up with that bond, which we know is a 1,4-glycosidic linkage because this is now maltose. In order for those substrates to bind to the active site, they've got to get close. We need to have some kind of collision between the active site and the substrate so that those two can bind. These collisions occur randomly. There are some forces of attraction between the substrate and the active site. It helps to kind of pull that substrate into the active site. But for the substrate to get close enough for those hydrogen bonds to actually take effect, we do need to have some random movement. This random movement is due to particles dissolved in the water, which is why it's so important for water to be part of life. So for our substrate and our enzyme to both run into each other, for these particles to run into each other, we do need to have some movement. If I have this guy sitting here and this guy sitting here and they never run into each other, then no reaction is going to take place. We can actually get more collisions to occur more frequently if I increase the substrate concentration. I like to use, not just me, all of us scientists like to use the shortcut of these square parentheses to show we're talking about concentration. So this is a low substrate concentration, a medium substrate concentration, a high substrate concentration. The more substrate we have floating around, the more frequently the substrate is going to run into the enzyme, and therefore the activity is going to increase. We're going to have an increased rate of reaction. Interestingly, at some point, it doesn't matter that I have more substrate rolling around. My activity level levels off or plateaus because my substrate and my enzymes are colliding as quickly as they can. So we say that our enzyme is saturated. It is as full of substrate as it can be. Those enzymes are working as fast as they can. It doesn't matter how much extra substrate pieces are rolling around. Those enzymes are working just as quickly as possible. Another way that we can change the rate of enzyme activity is by changing the temperature. Notice here that as we increase the temperature, we do have a nice increase in the activity rate. This is because higher temperatures mean more movement of our particles. And if our particles are moving more frequently and more quickly, they're running into each other more often, we have more collisions, we have more reactions. We're going to get to some optimal temperature, and then notice that things fall down really, really fast, which might feel kind of weird. Like, if things are moving faster and faster and faster, we should have more and more collisions. Except remember that because enzymes are proteins, proteins at high temperatures can be denatured. Proteins can be denatured. What does that mean? Remember that denaturation is this disruption of the folding patterns of our proteins. Proteins have those 4 levels of structure: primary, secondary, tertiary, and quaternary. Primary structure is that sequence of amino acids. This isn't so much disrupted by temperatures unless they get really, really high, so high that we're breaking the amino acid bonds, the peptide bonds between the amino acids. More likely, we're going to disrupt the secondary, tertiary, quaternary levels of structure. So here we have this nice protein. Notice all of its alpha helices. We've got some beta pleated sheets. We have some of that 3-dimensional tertiary structure, that 3D folding. When we have super high heat, these guys wiggle and jiggle apart. They don't stay folded so nicely. The alpha helices kind of fall apart because they're just too hot, which means that that molecule is just wiggling and jiggling a little bit too much to stay folded up in its nice shape. Sometimes we say that our secondary structure melts, that the alpha helices, the hydrogen bondings, are melting. We're not really turning it from like solid to liquid melting, but we're definitely losing the shape of that protein. Another way that we can affect the rate of enzymes is by changing the pH. Once again, notice that we have this optimal pH, and if we go away from that optimal pH, we're going to have a decrease in the activity, the rate of reaction of the enzyme. The reason for this is because we have those hydrogen bonds holding our alpha helices, our beta pleated sheets, that secondary structure, also tertiary and quaternary structures. Hydrogen bonding is holding those together. Hydrogen bonds are these forces of attractions between something that's slightly positively charged and something that's slightly negatively charged. Something that has a low pH, an acidic pH, has lots of hydrogen ions floating around in it. Those hydrogen ions are going to be attracted to the negative parts of that molecule, which means that the force of attraction here is going to be broken because that hydrogen ion from the acid is going to be in the way. Same thing happens if I have a more basic solution. We're going to have more hydroxide ions floating around. Hydroxide ions are negatively charged. They're going to be attracted to positively charged or partially positive charged pieces of the protein, which means that this hydrogen bond is going to be broken. That secondary structure of a protein is held together only by hydrogen bonds. So if we're breaking those hydrogen bonds by throwing in some extra ions, we're definitely going to disrupt the shape of the protein, the shape of the enzyme, which means that it cannot do its job anymore. Here, though, notice that I have really low pH, so pH of 7 is neutral. The lower we go, the more acidic the solution becomes. The higher we go, the more basic or alkaline the solution becomes. Trypsin is an enzyme that's in our small intestines, super close to an optimal pH of 8. Pepsin here is closer to 3. That's really quite acidic. Pepsin, you might have already guessed, is an enzyme that functions quite nicely in our stomachs, which have a much lower pH, a more acidic environment. One more way that we can affect the activity of enzymes, we can change the rate, is by adding some inhibitors. This is showing us normal activity. I've got my enzyme, I've got my substrate. That substrate fits quite nicely into the active site of the enzyme and things happen. We can add a competitive inhibitor. The competitive inhibitor's shape is going to be similar to that of the substrate, which means that it's going to fit quite nicely into the active site of the enzyme. It literally competes for the active site. And if that inhibitor is bound to the active site, the substrate cannot bind to the active site, and so things don't happen as quickly as they did when we didn't have that inhibitor. Our other kind of inhibitor is a non-competitive inhibitor. The non-competitive inhibitor does not compete for the active site, which is why we call it non-competitive, but instead it's going to bind to some other piece of the enzyme. We're going to call that other spot the allosteric site. So that non-competitive inhibitor binds to the allosteric site. When it binds here, it causes a shape change to the active site of the enzyme. And so now, even though there's nothing preventing the substrate from getting there, it's the wrong shape, and so it cannot bind. Nothing is going to happen. This graph is showing us how substrate concentration can affect how much a non-competitive inhibitor and a competitive inhibitor might change the rate of reaction. So let's say that I've got just a little bit of substrate here. The non-competitive is going to slow things down, competitive is going to slow things down, and then here's my normal enzyme working pretty quickly. Notice that the more substrate we have, the closer the competitive inhibitor and the normal enzyme activity become. This is because if I have like 10 pieces of inhibitor, but 10,000 pieces of substrate, of course, the substrate is going to get to the enzyme more frequently, and then we'll have some pretty big activity happening. For our non-competitive inhibitor, because the non-competitive inhibitor is not trying to get to the active site, it can have a big effect on the enzyme even when I have lots and lots of substrate. This shape change is going to prevent activity no matter how much substrate is rolling around. We actually put a lot of enzymes to work for us in technology. We didn't always think that this was going to be possible. Back in the late 1800s, there was this idea of vitalism. Louis Pasteur of spontaneous generation fame said that alcohol fermentation can only happen if we have cells present to carry out that fermentation, because it is an organic chemistry, biological function that can be done only by biological things like cells. Then the Buchner brothers here used a yeast extract, so not actually yeast cells, but an extract of yeast to make some alcohol. And for that, Edward won a Nobel Prize. Today we use more than 500 enzymes without any cells in lots and lots of commercial processes. We will often immobilize these enzymes that we use in technology for quite a few really important, wonderful reasons. One is that we can control the conditions that those enzymes are in. We can prevent their denaturation. We can keep them just the right temperature. We can control the rate of reaction by controlling those conditions. We can also greatly increase the concentration of the enzymes by mashing them all together, by immobilizing or gluing them onto some surface. Here are some different ways that we can immobilize our enzymes. We might stick them onto some kind of support. We might glue them together. We might trap them in these matrices. We might encapsulate them in some kind of membrane. Regardless of what we're doing, we can definitely mash those enzymes together super, super closely, increasing the concentration of them, which means that they can do a lot of work in a very small area. We also, by mobilizing the enzymes, can more easily separate the products from the enzymes. That also allows us to reuse our enzymes. Here's an amazing example of how we can use enzymes. So these beads have immobilized lactase enzymes on them. We're going to take regular old milk that has milk sugar, lactose in it, and we're going to pour the milk into this flask, and it's going to roll around the beads as it drips through the flask. And as it rolls around these beads, the milk sugar, the lactose, is going to be broken down into glucose and galactose. People who are lactose intolerant do not make lactase enzymes anymore, and so too much lactose in their bellies, in their small intestines and their large intestines, is going to lead to some discomfort in those bowels. Lactase will break that lactose down into glucose and galactose, which doesn't cause any of that discomfort. So we end up, after that milk drips, drips, drips past all these beads with all that lactase enzyme, the lactose is turned into glucose and galactose, and we get this milk that has lots and lots of glucose and galactose, but no lactose anymore. And that makes it safe for people who are lactose intolerant to drink and have cereal and cheese and all the yummy things that come with that. And on that note, my friends, we have arrived at the end of our lecture. Good work today. We talked about how proteins can be denatured. We can denature them with temperature, with pH. We talked about how the function of a protein is related to its structure. Structure-function correlations are so important in biology. Our enzymes are special metabolic proteins. They carry out chemical reactions. They actually reduce the activation energy of some chemical reactions. They do that by having substrates bind to their active sites. We can change the rate of enzyme, oh my gosh, that's still an of, if the rate of enzyme activity by changing substrate concentrations, by changing temperature, by changing pH, and by adding some different kinds of inhibitors. And of course, we use those enzymes quite a bit in technology. Great work today.